Hey everyone, we're going to be studying Mendelian genetics, and we'll be doing different types of genetics problems, including Punnett squares that follow normal, what we call Mendelian rules, and then other things that we've discovered since Mendel's time where other genetics rules apply. So in your notes, you can find them on Google Classroom. One of the slides will have the readings for the chapter kind of broken up for you, if that helps you do a little bit each day. So when we talk genetics, we often talk about a man named Gregor Mendel, and even honor him by talking about Mendelian genetics or Mendelian rules of genetics. You'll hear that a lot. But Gregor Mendel was an individual that did live in the 1800s and wanted to study science. Back then, if you want to study something and maybe you didn't have a lot of money, a good way to get those opportunities was to join the clergy. So Mendel became a monk and ended up working in the abbey in the garden and getting a lot of opportunities because that's where the money was at the time, was with the clergy, with religion. So he got to study under people and pursue what he wanted to pursue, and then ended up doing inheritance experiments while he was working in the abbey garden. He ended up doing his passion, basically. And when you hear Mendel, you often hear about pea plant experiments. It's a good thing because peas you can do genetic experiments with, but you can also eat the product when you're done with it. So it worked out pretty good. So we honor Mendel still because of different things. First of all, he used experimental method. He had good quantitative and qualitative analysis, took good notes, and collected data, had large sample sizes, which is great, and excellent scientific method. Maybe one of the best things to think about here is that he spent 2 years planning what he was going to do, planning his research and experiments, getting it ready. It's hard to think to spend 2 years doing that before the actual action starts, but that's what he did. So he ended up looking at different traits with pea plants. Back in his time, the idea was called the blending hypothesis, where you look at an individual, whether it be another animal, human, or something else, and the individual tends to look like a blend of the parent's traits. So people just thought that that's what happened. That individual who was born from a couple would have a blend of that couple's traits. So it's called the blending hypothesis. And so Mendel set out to test that. And he used different traits with pea plants. As you can see, there's a bunch of things that you can choose here. We're going to use purple and white flower or blossom color for our first discussion, but you can use other things like, did you know that pea plants come with different seed colors, different seed texture, whether it's round or wrinkled? The pods are different colors. The pods have different textures, whether they're inflated or constricted. The flowers can be found kind of in the middle of the stem. Those are called axial or terminal when they're on the end. And also the plants can come in tall and short. And if you look at his data here, just quickly with this experiment, you can see nice large sample sizes. Here we've got over 6,000, over 5,000, in the hundreds. Remember, in science, it's always good to have a large sample size. So we can credit Mendel with that. So let's talk about his experiment to learn about inheritance and test that blending hypothesis using the purple and the white blossom colors of the peas. A little review of plant reproduction. Plants have male and female parts. If you look at the plants here, there's this middle thing here, right there, and you can see it over here. It's kind of tan. That's a female part. And down that tube are the ovaries where the eggs are produced. These on the end here with the little powder coming off of it, that's where pollen is coming from. Pollen is like sperm, and so it's really fine and can flow through the air, travel on bees and animals. And the idea is to get that pollen, aka sperm, onto the top of the female part so that it travels down that tube and then fertilizes the eggs that are inside there. And restoring, going from haploid, both of the gametes haploid, and restoring the diploid condition. So that's what a plant is like, and you can control this yourself by, if you don't want this plant to self-fertilize itself, because that pollen only has to go from here to there, if you don't want that to happen, you can just simply cut them off, and then you just have the female part there left. The idea, remember, with sexual reproduction is to have diversity. So if plants are just fertilizing themselves, you're not going to get a lot of diversity that way. So hence we have wind and bees and animals and different things that can help to spread that pollen around a little bit and ramp up the diversity in the species. So Mendel spent 2 years getting this stuff ready, and they noticed that these traits for blossom color came in 2 varieties, white and purple. So obviously you would predict then, if the hypothesis of blending is correct, that the offspring would be just a blend, kind of maybe a light purple, if that was the case. So he spent 2 years getting this ready so that the white ones only ever had white blossoms. So he would separate those into probably one part of the garden, and then the purple ones in another. That way they wouldn't cross-pollinate each other. And so for 2 years, those pea plants only ever produced their own color. Purple over here, white over there. And then after 2 years later, we're gonna try out the experiment. So some words there. True breeding means that they only produce their own color. Today we would know that word as homozygous, if that word rings a bell for you. We could also call it pure breeding. So the pure breeding with white and pure breeding with purple, or they're both homozygous for sure for their trait, because that's all they've ever been producing for the last couple years. So we're going to call that the P generation. That's our parents. And then we're going to cross-pollinate them and get what we call the F1 generation. F actually stands for the word filial, but you can think of it like the first generation of offspring. So here's our F1s here, and when he finally allowed them to cross-pollinate, the result of the F1 was all purple. And the purple was the same color as the P generation, as the parents. Not a lighter color or a blended color or spots or anything like that. So now the question is, where did the white go? That's interesting, because I think we falsified that blending hypothesis, right? So then Mendel questioned it further and decided to take this a step further and create an F2 generation by letting these F1 pea plants self-pollinate. And so, can you guess what happened in the F2 generation? Let's take a look here. So here's the beginning of our experiment. We've got the P generation with our purple and white blossom plants. 100% of the F1s were purple. So we called those hybrids. Today we would also call them, remember the word heterozygous, because they are carrying the genes from both of the parents. Also keep in mind, in the 1800s, they did not know about genes or DNA yet. So Mendel is doing all of this without even knowledge that DNA or genes exist. So here's what happens in our F2 generation. After they self-pollinate, 75% of them still look like the parent, who is purple. Again, not lighter color. It's the same, whatever color purple the parent is. But the white shows back up. It is back. And 25% of them were white. So with our numbers here, we can get a little quantitative analysis with 75% and 25%. We can also do fractions here with 3/4 or 1/4. And we can also do ratios of 3 to 1 ratio. So we have falsified our blending hypothesis. This is called a monohybrid cross, by the way. We are looking at only one trait here, and so mono for one, monohybrid cross. And so here's what Mendel concluded from his experiments. So first of all, traits come in alternative versions. So we have purple and white for flower color. Flower color is our trait. So the different versions that flower color comes in, he called those alleles, and we still use that word today. Alleles are different versions of a trait. And we can also say today alleles are different versions of a gene, because today we know what genes are. A gene, remember, is a sequence of DNA that codes for a protein. And so an allele just has a little bit different sequence that causes the trait to vary. So different alleles vary in the sequence of nucleotides, those are the A, T, C's, and G's, at a specific locus of the gene. So here's our homologous chromosomes. Here's where the gene for flower color is located on those chromosomes. So they come in different alleles, so that must mean there's a little bit different sequence of the nucleotides when we compare the two. And then the location where those alleles are located is called a locus. That's what scientists use for their terminology. Let's get used to using the correct terminology for all of this. Here's the next thing that Mendel concluded. So besides having the word alleles to explain the different versions of traits, for each characteristic, an organism will inherit 2 alleles, one from each parent. This is for a diploid organism. Diploid organism would carry 2 of each of those chromosomes, so we would find the alleles on each of those chromosomes, so 2 copies, one from each parent, one from mom, one from dad. What's the advantages of this? Well, I guess one advantage might be that if one of the alleles that you get from a parent is mutated, hopefully the other one is good and functioning. Here's our next finding. Some traits have the ability to mask or cover up others. So the purple and white flower colors are separate traits, separate alleles. They do not blend. And one of them in that F1 generation is the one who showed up, and you couldn't get the other one until the F2 generation. So the dominant one then would be the purple, and it would be the one that's expressed when you actually have that allele. The other one gets covered up for a generation. That's called the recessive allele. So purple would have been the dominant and white would have been the recessive. Often you can think of as the dominant allele as the functional protein, but not always. There's exceptions to that, and it again masks the other alleles. And then the recessive allele as the malfunctioning protein or the absence of the protein. But not always. Again, there's a lot of exceptions to those rules. With the white flowers, you can think of it as an absence of pigment, whereas the dominant allele is the presence of the purple pigment, the functional purple protein pigment. Sometimes they call the dominant allele the wild type. This is the allele producing the functional protein, and the one you normally would see out in the wild in nature often. And then the mutant one could be thought of as the allele producing the malfunctioning protein, or possibly the recessive allele. Again, exceptions to many of these rules, but this is what Mendel is finding now at the time, again, knowing he has no idea about DNA or genes or any of that modern genetic stuff that we know of today. Here's the next thing that Mendel figures out. There's a difference between how an organism looks and its genetics. How an organism looks, we're going to use the word phenotype. It's the physical trait or the physical characteristic. The genotype is the organism's genetic makeup, their gene combinations. All right, so often we use letters to symbolize the genotypes when we do genetics problems. And then we use capital letters for dominant genotype and lowercase letters for recessive genotype. And then the phenotype is the result of the genotype. So when we cross individuals, such as in a test cross, we use these letters. So for purple, we could use the word P. We don't have to use P. We can use any letter that we want. But since purple is the dominant allele, we can go ahead and do that. When using P's, though, I'll caution to make sure you make your big P's big, make them look big, in uppercase, and make the smaller ones, the recessive alleles, make them look lowercase as well. So the recessive allele then would be the little p. Use the same letters for dominant and recessive when we're using what we call regular Mendelian rules of complete dominance. So purple is completely dominant over the white. We'll give purple the big uppercase P, and then we'll give white the lowercase p. So the true breeding purple flowers that we started off with would have had a genotype of big P, big P. The true breeding white flowers would have had a genotype of little p, little p. Both of them are homozygous for their traits. The purple ones are homozygous dominant, and the white ones are homozygous recessive. So here we can see them in a cross, and then we think about, well, what alleles can the parents provide to their offspring? Well, if one parent is big P, all they can give is a big P. No surprise there. So bring the big P down, and the other parent can only give a little p, bring that down. So our F1s are all big P, little p. They're heterozygous. They have one of each of the alleles. Because the purple is dominant over the white, that's the phenotype that we will see, even though the genotype does show that they're carrying that white allele, but it's just being masked by the dominant one. So here we have all those words written in here with our purple and white as the phenotype of the parents to begin with. We have their genotypes written here, big P times little p. All of the F1s are the same, heterozygous, big P, little p, 100% of them. And then when we self-pollinate them and get the F2 or second filial generation, then 75% of them would have been purple phenotype and 25% would have been a white phenotype. Now with the white ones, you know their genotype. It's little p, because whenever the dominant one is present, then it would be purple. So the only way to be white is to be little p. However, we have a question mark with what the genotype is for these F2s who are purple. We don't know. We know they all at least have one big P, but we don't know what the other letter is, what the other allele is that they're carrying. So a Punnett square can help us figure out what the probability is of what their genotype is. So here's what our Punnett square would look like. Punnett is capitalized because it's named after someone. So our F1 generations were all big P, little p, 100% of them. So if we cross a big P, little p with a big P, little p, and then put them into our Punnett square like this, we put one parent here. It can be the male or the female, it doesn't matter. But big P, little p. Over here on the other side, either again the other individual, big P, little p. And then we fill in the boxes. Bring your big P's down. So big P in this square. Big P over, little p there. Same with this box. And then the little p's here in the other corner. Again, I caution using p's because sometimes a big P is hard to tell versus a little p with people's writing. Even when it's typed, sometimes can be difficult. But just be careful about that. And also, I caution you, when you're writing these, always write the dominant allele first. So for our heterozygous individuals, it's big P, little p. Don't write. So we can say the probability is is that there's a 1/4 or 25% chance back here that these flowers are big P, and then there's a 50% or 1/2 chance, that they are big P. And then the 25% shows up in our Punnett square here, who are little p. Notice how the percentages for the genotype are different than the phenotype. So 75% purple, 25% white for the phenotype, but 25%, 50%, and 25% for the genotype. And then you can also use ratios like 1 to 2 to 1 and a 3 to 1 for the phenotypic ratio. So there's our words. Remember to use the word homozygous when they're the same alleles, big P, and little p. Big P is homozygous dominant, little p is homozygous recessive. When they have one of each allele, it's heterozygous. Big P. Always write the big letter first. So two organisms can have the same phenotype, but can have different genotypes, such as the big P and big P. For the dominant allele, you only need one copy of it, and you will display that or express that as your phenotype. So how do you determine the genotype of an individual with a dominant phenotype? How do we really know that those flowers were purple and what their genotype would be? So the answer is we can use a test cross. That means we're going to breed those purple flowers right here with something that we know for sure. What do we know for sure? Well, we know the white ones are all little p for sure. They're all homozygous recessive. So we're going to cross them and see what we get. And that should give us a better idea of the genotype of those F2 generation purple flowers. So if this is the case, we got our little p's, we know that for sure. But if this is the case, if those flowers were big P, then we would expect all of the F2 generation, or all of, the next generation to be purple. If they're big P, if they're heterozygous, then we're going to expect half and half. We're going to expect some white ones in the bunch. So that's how we can tell. This again is called a test cross. They do use this in breeding programs, such as with breeding cats and dogs and domesticated animals, things like that, and plants as well. So from this, then we get some laws of heredity. Mendel actually was able to come up with some laws of heredity. So the first one is called the law of segregation. When you hear the word segregation, think of the word separation. So this simply means separation. During meiosis, the alleles segregate or separate into separate cells or separate gametes. So basically, remember, if your genotype is big P, you can't give both of those big P's to your child. You can only give one in sexual reproduction because they're going to get the other one from the other parent. So which one can you give? Well, not a big surprise because you're homozygous, so you're going to give a big P. But these are both going to separate into different gametes. If you're little p, no big surprises there. You only have little p's to give. But if you're heterozygous, big P, then half of your gametes will have the big P in it, and the other half will have the little p. So the law of segregation. Your alleles will separate into different gametes. This happens during metaphase 1. And like it says, whoa, Mendel didn't even know DNA or genes existed at the time. So as I said before, the experiment that we did was a monohybrid cross, only looking at one trait at a time. And monohybrid crosses can help show us Mendel's law of segregation. So when we set up a Punnett square, I should say, each of these boxes represents a different gamete. And when we're putting them into those boxes, that's symbolizing how they're separating into the different gametes. We can also make this more interesting by doing what's called dihybrid crosses as well. Di means two, so that means we can follow two traits that an individual has at a time, such as what Mendel did with both seed color and seed shape. Look at the picture. Some of the seeds are round, some of them are wrinkled, some of them are yellow, some of them are green. From dihybrid crosses, which we will study a little bit later, he was able to figure out another law, and that law is called the law of independent assortment. And we've looked at this thoroughly when we've studied meiosis before. We know this happens during metaphase, and it all depends on which side, remember, that the homologous pairs get on. And so it's all done randomly, independent assortment, or random mixing of the different chromosomes that the parent are providing. And we'll be looking at that a little bit later, a little bit more in depth when we do dihybrid crosses. In summary, we want to give Mendel some credit here for doing what he did in the 1800s without even knowing about DNA or genes at the time. He chose wisely. Maybe some of it was due to luck as well, but pea plants are good for genetic research. They're available in many varieties. They have distinct heritable features that we can see, like flower color, seed color, and seed shape. He had strict control over which plants mated with who. He spent time getting that ready, remember, making sure they were true breeding for their traits. Pea plants can self-fertilize, which can help us get that F2 generation. And so Mendel could cross-pollinate plants and move pollen easily from one plant to another. You can use a little brush and do it on your own if you like. The actual traits that he was using with the pea plants were relatively simple genetically, meaning those characters or traits were controlled by a single gene. We know today there's more complex things going on with some traits, but the ones that he chose with peas, luckily, were only controlled by a single gene. Had they been controlled by more than one gene, we could have gotten different results, and he could have been thrown off by some of those results that we know of today. And the traits that he used, one was completely dominant over the other. Again, we know of other rules for that exist as well. Sometimes one gene is not completely dominant over the recessive gene, but for the ones that Mendel chose, like the purple and the white flowers, that tended to be the case. He took, again, good data, good journaling, good quantitative data as well, and then he actually published his findings, which is very important in science to share your findings. However, because not a lot was known about this at the time, it kind of got ignored. And there was another person making research known at the time. His name was Darwin, and I think maybe we were more focused on his because his might have been a little more controversial, and then we ignored Mendel. However, if those two guys would have gotten together and pooled their research, genetics and inheritance can help explain how evolution works. It would have probably made things a lot easier and moved the science along a lot more quickly. So remember, if you have any questions, let me know.